One of the experiments that's just delivered to the station inside that SpaceX Dragon cargo vehicle is investigating how cells adapt to stresses they encounter from being in the low Earth orbit weightless environment. It's called the Advanced Plant Experiments 2, or APEX 2, and this morning we're going to learn about it from Principal Investigator Dr. Timothy Hammond of the Durham Veterans Affairs Medical Center and Duke University School of Medicine in Durham, North Carolina. Dr. Hammond, thanks for joining us on Space Station Live.
Good morning. It's a pleasure.
That's great to have you here. It's hard enough to figure out why entire organisms react as they do, but tell us why you are focusing on the cellular level, if you will.
Well, we have the privilege of flying yeast, which actually have about half the same genetic makeup as a person. And our long-term goal is to repurpose drugs. Getting new drugs on the market is expensive, and finding one without side effects is difficult. And in the VA, we have commonest diagnosis is type 2 diabetes, and the commonest medicine for that is metformin. And what we have found is patients that take metformin for diabetes are protected from certain forms of cancer. Modeling those cancers is difficult on the ground because in the middle of the cancer you have very little gas exchange. And it's exactly the same in space. You have very little convection. The gases don't move like they do on the ground. So we think space is a very good way to model the inside of a tumor and to look for new drugs.
Well, let's see. You're actually using brewer's yeast for this experiment, I understand. What makes that appropriate for this type of research?
Brewer's yeast is great because it's simple. It's simple to grow. We know every gene that it has, and its control systems are very simple. And it has some special enzymes that make it very easy to do sophisticated molecular biology and knock genes out. So we actually have a series of genes, a pool of genes, a pool of strains in which one gene is knocked out of every strain, a different gene, and it's been replaced by a piece of DNA that we know its sequence. And when we then grow the yeast under a pressure, in this case space, or space with and without a drug, and that way we can count those barcodes, as we call them, that we put in, and know what gave us survival advantage and disadvantage when the samples come back.
How does the experiment actually operate on the station in terms of autonomous, crew involvement, both?
Well, actually, this time what we've done is we've put, if you've ever seen a fluorescent jellyfish that glows green,
right,
the protein that makes it glow green has been isolated, and we have its DNA, and we've put that in front of every gene in the genome as well. So it's spotted. Each strain reports on when a gene is transcribed, it also transcribes that protein that makes a green fluorescence. So we have plates that have 384 different strains, each one reporting a different gene. And over 10 plates, we can get the whole genome. And there's actually a fluorescent plate reader on station. We put one plate in at a time. We measure the gene expression every 10 minutes for 18 hours, and we get the entire ballet of how yeast respond to space.
That's amazing how that is planned to operate. How are you going to be able to tell basically how the cells respond to the stresses from the orbital environment?
Well, of course, we have ground controls for comparison, and our Canadian collaborators who actually do the spotting, Canada has fantastic robotics. Just like they made the Canadian arm for the International Space Station, they have some very, very good robotics. And they just published a paper in Science last week in which they look at 3,700 different responses to stresses and drugs on the ground. So that gives us a fantastic reference library. So we really understand what's unique about space and what we can exploit in space that we can't do on the ground.
And of course, we'll talk about your goals to figure out. Are they to figure out which plants grow best in space, or how to genetically engineer plants so that they can grow well in space, or might even the findings be applied to cells of people who will actually be in space?
Well, actually, we're interested in helping the astronauts, of course, but we're interested in veteran patients in VA hospitals and Americans in American hospitals. and in international countries. What we want to do is repurpose drugs, and we're interested in whether the lack of convection in space, the lack of gas movement, gives us a good model for tumor agents, for anti-cancer drugs that are better than on the ground. So what we will bring back is the knowledge of which pathways attack tumors so that we can use those drugs or even design better drugs on the ground once we know the pathways involved.
Well, I think you just answered my last question, but the unique, I guess, environment of space offers you actually I assume you can have direct interaction with the crew as a principal investigator, right?
We do. We, of course, have to go through the CapCon like everybody else, like you see on TV. But yes, we can. And we try to design an experiment that is fairly automated, so we don't take it. So we have abundant crew time when we need it, but we try not to overuse that privilege.
Well, we're certainly looking forward to hearing all about it even further, and we really appreciate you joining us today on Space Station Live, Dr. Hammond. Thanks a lot.
Our pleasure. Thank you so much, sir.